Hello everyone. It is a great honor to share a study from Dr. Shaogui Wan's group at the Gannan Medical University. This study introduces a bioinformatics pipeline, named as ECCFP, for eccDNA identification from long-read sequencing data. eccDNA, or extrachromosomal circular DNA, is a circular DNA molecule that exists independently of chromosomes. It plays a versatile role in pathological or physiological processes, such as in tumorigenesis. Currently, eccDNA detection usually relies on short-read next-generation sequencing (NGS) technology, but it is challenged for identifying eccDNA derived from tandem repeat regions. Recently, long-read third-generation sequencing technique enables direct identification of complete sequences, offering promising opportunities for eccDNA detection. However, the bioinformatics analysis for eccDNA identification from long-read sequencing data still have many challenges, such as high false positive and low position accuracy. ECCFP uses all consecutive full passes from individual reads for candidate eccDNA identification and consolidates candidate eccDNAs to generate accurate unique eccDNA. This strategy boosts eccDNA detection and reduces false positives. ECCFP performs well across datasets in sensitivity, accuracy, and running efficiency. By the way, ECCFP is a fully open-source pipeline and user-friendly. ECCFP contains 4 steps. Firstly, quality control and classification of raw alignments, with retention of ghost sequences that current methods often overlook to maximize data utilization. Secondly, extract candidate eccDNAs from consecutive full passes. Thirdly, consolidate candidate eccDNAs with Boyer-Moore majority voting to generate accurate unique eccDNA. Fourthly, output genomic coordinates, consensus sequences, and variant information. Taking HepG2 cell sequencing data as an example, excluding ghost sequences would discard nearly 40% of the usable data. This study showed that ghost sequences also exhibit high sequencing quality. ECCFP successfully identified 1,613 new eccDNA from these ghost sequences, which were validated by Sanger sequencing. Taking a certain region from chromosome 12 as an example, ECCFP identified 7 candidate eccDNAs from individual sequencing reads. Using the Boyer-Moore voting algorithm, ECCFP merged multiple candidate eccDNAs into a unique eccDNA with precise genomic coordinates, which was confirmed by Sanger sequencing. To evaluate the performance ECCFP, for artificially synthesized circular DNA were spiked into HepG2 cell samples. The sequencing results showed that ECCFP fully identified all spiked-in circular DNA with accurate junction position validated by Sanger sequencing. Furthermore, the number of eccDNA identified by ECCFP highly correlated with the spike-in copy numbers. Simulated datasets based on real rolling circle amplification were used to further evaluate ECCFP performance. Across 4 pipelines with varying depth and read type proportions, ECCFP demonstrated stable and superior performance in very depth simulated datasets. These results support ECCFP's broad applicability and robustness. Finally, ECCFP was evaluated using 29 public datasets from diverse species. ECCFP showed high accuracy and run efficiency across sample types, which identified more eccDNAs and had a shorter runtime than other pipelines. Conclusively, ECCFP uses all consecutive full passes from individual reads for candidate eccDNA identification, and then consolidate candidate eccDNAs into accurate unique eccDNA by Boyer-Moore voting algorithm. ECCFP demonstrates significant advantages in sensitivity, accuracy, and runtime efficiency. As an open-source tool, ECCFP may provide a promising tool for eccDNA study.